class of compounds of amines. Now we have the functional groups. So first we have hydroxyl, which is in the class of compounds of alcohol. The structural formula is -OH. An example of it would be ethanol, like wine or beer. Then we have a carbonyl, which is polar, as well as the hydroxyl. The class of compounds is aldehydes and ketones. The structural formula is -C. On top is double bond to O, and then -C on the other side. An example is formaldehyde or acetone or nail polish remover. The third functional group is carboxyl, which is polar and acidic in the class of compounds of carbolic acids. Then we have -C with a double bond to O and a bond to OH. An example would be vinegar and citric acid. The functional group amino is polar and basic and is in the class of compounds of amines. The structural formula is -N, -H, -H. An example would be ammonia or the rotting fish stink. Then we have sulfhydryls, which are slightly polar in the class of compounds of thiols. Structural formula is -S, -H. An example would be rubber. And then we have phosphate, which is polar and has a negative charge. It falls in the class of compounds of organic phosphates. The structural formula is -O-P-O negative. On top, there's a -O negative. Below, there's a double bond to O. And an example of this would be ATP and DNA. Now we have biochemical reaction. So an anabolic reaction the definition is involve the joining of smaller molecules into larger ones, and a catabolic reaction is the breakdown of large molecules into smaller ones, releasing the energy contained in the chemical bonds. Now we have the biochemical reactions. So we have neutralization, which is neither anabolic or catabolic. The definition of neutralization is a chemical reaction between an acid and a base producing water and salt. An example would be baking soda and vinegar, which combine to make carbon dioxide, water, and salt. Then we have the oxidation-reduction reaction. So lose an electron is oxidized, gain electron is reduced. It is neither anabolic or catabolic, and the definition is a chemical reaction that involves the transfer of electrons from one substance to another, also called oxidation-reduction reaction. An example would be combustion of propane in a gas barbecue and cellular respiration. Then we have condensation, which is an anabolic reaction. The definition is a chemical reaction that results in the formation of a covalent bond between two molecules with the production of a water molecule. An example is the digestion of a polysaccharide, because it breaks down the macromolecule into simpler sugars, and glucose molecules join together to make starch. Then we have hydrolysis, which is a catabolic reaction. It is a chemical reaction that results in the cleavage of a covalent bond with the addition of a water molecule. An example of this is when the body uses glycogen by breaking it down into individual glucose molecules. Then we have carbohydrates. So among the most organic molecules on Earth contains carbon, hydrogen, and oxygen in the ratio of 1:1. The general empirical formula for all carbohydrates is CH2O. Okay, skip that. The function of a carbohydrate is that it's used by organism as a source of energy. It's used as building materials, and it's a cell surface marker for cell-to-cell identification and communication. Then we have monosaccharides, which are simple sugars consisting of one sugar unit. It's a straight carbon chain or ring with a hydroxyl group attached, distinguished by the number of C atoms in the carbonyl group. It is polar because of the high proportions of hydroxyl/carbonyl group, so it easily dissolves in water. An example is glucose or fructose. Then we have structural isomers, which notice that glucose and galactose and fructose are all 6-carbon sugars, same molecular formula, however the spatial arrangement varies. Therefore, they are said to be isomers. The difference in shape leads to physical and chemical properties, so they're treated differently in the body. A stereoisomer, so if the hydroxyl group is located below the plane of the ring, it is alpha glucose, and if the hydroxyl group is above the plane of the ring, it's beta glucose. Then we have disaccharides, which are two simple sugars bonded together. They are attached by covalent bonds with a glycosidic linkage between two hydroxyl groups, formed through condensation reaction. Common disaccharides are lactose, sucrose, etc. Then we have polysaccharides, which are complex carbohydrates. They are long chains of monosaccharides, several thousand. The lower solubility. They have lower solubility in water due to extreme length. And examples are storage polysaccharides like starch and glycogen, and structural polysaccharides like cellulose and chitin. Then we have starch, which is the main storage molecule in plants. Potato starch is a combination of amylose, which is straight chain 20%, and amylopectin, which is a branched 80% chain polysaccharides. It is insoluble in H2O. And animals use enzyme, example amylase, to break down amylose and amylopectin into individual glucose molecules. This is used in cellular respiration to extract energy. Then we have glycogen, which is energy storage molecule in animals. It's used to store excess glucose. Small amounts are stored in the liver and muscle and are used for energy during bouts of physical exercise. Then we have cellulose, which is the primary structural polysaccharide in plants. It's a component of cell walls. It's a straight-chain polymer of beta-glucose held together through a 1 to 4 glycosidic linkages. It is formed by alternating orientation of glucose subunits. It has alpha orientation in starch and glycogen and a beta orientation in cellulose. There is alpha orientation of a carbohydrate in starch and glycogen, and then beta orientation of carbohydrate. Then we have cellulose, which is resistant to starch breaking enzymes due to irregular orientation. It is a rich source of energy for organisms that can digest it. It is part of a well-balanced diet, so roughage, and what it does is pass through undigested in humans, scrapes large intestine, secretes mucus, lubricates feces, and aids in elimination. Then we have chitin, which is found in the hard exoskeleton of insects and crustaceans in the cell wall of the fungi. It is composed of a modified cellulose material and is a nitrogen. Now we have lipids. Lipids are long-term energy storage molecule. They contain C, H, and O, and they contain more C to H bonds and fewer O to H bonds compared to carbohydrates. They're hydrophobic molecules and insoluble in water, but soluble in other nonpolar substances. The functions are long-term energy storage used for building membranes and other cell parts and chemical signaling molecules. Lipids can be divided into 4 families: fats, phospholipids, steroids, and waxes. Fats are the most common energy-storing molecules. They store twice the chemical energy of carbs and proteins. For animals, excess carbohydrates are converted into fat molecules as droplets in the cells of adipose or fat tissue. Triglycerides, the most common fat in plants and animals, and they contain 3 fatty acids and 1 glycerol molecule. They can be saturated, so all single bonded carbons, or unsaturated, which contain double bonds. Then we have the phospholipids, which are a lipid molecule that composes cell membranes. It is made of 2 fatty acids, 1 phosphate, and 1 glycerol. When they're added to water, the phospholipids form spheres called micelles, which have hydrophobic heads, so they orient towards water, and hydrophobic tails, which orient inwards away from towards each other. Then we have steroids or sterols, which contain 4 fused hydrocarbon rings and several different functional groups. They are chemical messengers in the body. Then we have waxes, which contain long-chain fatty acids linked to alcohol or C rings. They're hydrophobic with a firm, pliable consistency and are ideal for forming waterproof coatings. Then we have proteins, which are the most diverse and most important molecules. It is genetic information in DNA that codes for the production of proteins and nothing else. They accomplish a variety of tasks, so there are structural building blocks like keratin found in hair and functional molecules like hemoglobin, which transports oxygen. Cells contain thousands of different proteins, each performing a specific task in 3D structure and directly related to its function. So, structure in R group. There are 20 different R groups found in living organisms. Depending on the nature of their side chain, amino acids may be polar, hydrophilic, nonpolar, hydrophobic, or charged, acidic, or basic. There are 8 essential amino acids, which must be obtained from diet, and 12 non-essential. First, we have polypeptides, which is an amino acid polymer constructed through protein synthesis. Amino acids are bonded together via a peptide bond, and is from a condensation reaction. So the sequence of amino acids determine the polypeptide's 3D shape, which also determines its functions. There are 4 levels of structure: primary, secondary, tertiary, and quaternary. Primary and secondary are linear, which are fibrous narrow strands. Tertiary and quaternary are globular and have a rounded, compacted shape. So primary is a sequence of amino acids determined by genes and DNA. Nucleotide sequence determines amino acid order. Change in one amino acid could alter the final shape of the protein. Then we have secondary, which is polypeptide coils and folds, and the amino acid grows in length. In some regions, 8 bonding occurs between carboxyl and amino and other intermolecular forces. The A helix is a H bond every 4 peptide bonds in a pleated sheet. Alpha helix and beta pleated sheets, which are H bonds between adjacent strands. Then we have the tertiary, which are the strong forces of attraction and repulsion between polypeptide and its environment, force it to undergo additional folding. They may also use chaperone proteins to help. Amino acids with hydrophobic R groups congregate away from water. The tertiary structure is stabilized by R group interaction, so H bonds, ionic bonds, proline kinks, and disulfide bridge. So how it starts is with a hydrogen bond, proline kink, hydrophobic and van der Waals interaction, disulfide bridge, ionic bond, and then the polypeptide backbone. Then we have quaternary, which is 2 or more polypeptide subunits which are joined together to form a functional protein, so example, hemoglobin. Then we have factors affecting shape or folding, so the primary structure, there's potential for intramolecular forces, temperature also affects it, pH, and ionic concentration. Then we have denaturation. So denaturation is a change in a protein's 3D shape, which also means a change in its function. Denatured proteins do not carry out functions, and they may return to function if the denaturing agent is removed. The protein will be destroyed if the primary structure is altered. Denaturation is dangerous because it increases in the body. So for a fever, it increases in the body temp and denatures enzyme, which can lead to seizures. And it's useful for pickling vegetables, heating your hair, or the digestive enzyme gastrin, which works at a pH of 2 in the stomach and will denature in small intestine pH 10. Then we have nucleic acids. So nucleic acids are informational macromolecules. They store hereditary information that determines the structural and functional characteristics of an organism. There are only molecules that can copy themselves, so they allow the organisms to reproduce. There are 2 types of nucleic acid: DNA, which are instructions for creating an organism, and RNA, which reads the info in DNA and transports it to the protein-building apparatus of the cell. DNA and RNA are nucleotide polymers. The nitrogenous bases are adenine, guanine, thymine, and cytosine, and in RNA, thymine becomes uracil, so there's no thymine found in RNA. So forming a polymer of nucleotides is often referred to as a strand. A phosphodiester bond occurs between adjacent nucleotide subunits, between the phosphate group on the nucleotide and the hydroxyl group on the sugar on the next nucleotide. And there's also a sugar phosphate backbone. Then we have RNA versus DNA. So there are ribose sugars found in the DNA. There's a sugar phosphate backbone, and there are also the nitrogenous bases, so RNA is single-stranded, DNA is double-stranded with they go opposite ways. And then we have the DNA structure. So DNA runs anti-parallel to each other, so they run in opposite directions of each other. There are purines, which are double rings, but then they will always bind with a pyrimidine, which is a single ring. So adenine and guanine have 2 rings, and then cytosine and thymine have 1 ring. So adenine bonds to thymine, guanine binds to cytosine. And Then we have the cell parts and their function. So the cell wall is a rigid but flexible structure made of cellulose. It acts as a pressure vessel, preventing overexpansion when water enters the cell. It is found mainly in plant cells, but also in fungi, algae, and some bacteria. Then the cell membrane. The entire cell is covered by this and is mostly composed of phospholipids. It is selectively permeable and controls the entry and exit of various chemicals in and out of the cell. The flagella, which is the tail-like projection that helps the cell move. Cilia is slender protuberances that project from the cell body. They can aid in motion or serve as sensory organelles. Mitochondria provides the cells with energy by producing ATP through cellular respiration. Chloroplast captures light energy and so is involved in photosynthesis. And then cytoplasm, where most cellular activities occur, such as metabolic pathways, including glycolysis, and processes such as cell division. It is also the space that contains the organelles. The ribosome, which makes proteins out of amino acids. Lysosomes, which break down worn-out organelles, viruses, or bacteria and food particles. It does this with digestive enzymes in an acidic environment. Centrioles, which are critical for cell division and the organization of spindle fibers and mitosis, only found in animal cells. The smooth endoplasmic reticulum, which is a series of canals that carry materials throughout the cell and help synthesize lipids such as steroids. Then we have the rough endoplasmic reticulum, which is where many ribosomes are attached, so it helps synthesize proteins. Vacuole, which is a fluid-filled storage space containing water, sugar, minerals, or enzymes. Their function varies depending on the type of cell. Golgi bodies, or apparatus, which is a structure that stores and packages proteins such as enzymes and lipids. Nucleus, which is the control center of the cell that contains the DNA. The nucleolus, which is a spherical structure of the nucleus that makes ribosomal RNA, and then chromosomes, which is the name for the thread-like structures that contain genetic information. Then we got the phospholipid bilayer or the fluid mosaic model. So A phospholipid is a phosphate head which is polar and two fatty acid tails which are nonpolar, and they form a layer, so head towards the water, protecting the tails, which separates internal environment from external surroundings. We have protein channels which allow largely charged molecules to pass through the nonpolar part of the protein, so it transports. Then we have glycoproteins, which are a protein and a carb. So the carb reaches out to the environment to tell cells, example white blood cells, that's a part of an organism, and it also helps with cell recognition. Then we have the receptor protein, which is where a protein sits in a receptor. The bind changes the receptor's shape, which then changes the cell. Then we have the peripheral protein, which is involved in cell-to-cell signaling or in cell recognition. Then we have cholesterol, which provides stability to the membrane in a warm environment. So without it, it could fall apart if it gets too hot. And there's less cholesterol in a cool environment. Then we have the cytoskeleton, which helps cells maintain shape, structure, and organization, and it supports the cell, it's located in the middle of the cell. Then we have the types of transport. So there's passive transport, where no energy is needed. So for this, there is simple diffusion, which is small molecules that are moving from high to low concentration. And there's facilitated diffusion, which is the same as simple diffusion, but it uses a protein channel. It still goes from high to low concentration. Then we have active transport, where energy, or ATP, is needed. So there's transport proteins, which are the channels and molecules moving from low to high concentration. And then there's transport requiring vesicles, or membrane transport. So an example of this is exocytosis and endocytosis. Exocytosis is when things are exiting or exerting or moving out of the cell. The membrane kicks out molecules. Example is cell waste and cell waste. We have endocytosis, which is to enter or bring in, and the membrane brings in molecules. This then branches out into pinocytosis, phagocytosis, and endocytosis, and receptor-mediated endocytosis. And pinocytosis is like cell drinking, so the cells takes in fluid by pinching inward. And phagocytosis is cell eating, so the cells take in large things. And receptor-mediated endocytosis is using receptors to take in a large thing. The definition of diffusion. For diffusion, it is mainly small, non-polar molecules like oxygen, and they need to be able to slip through the phosphate heads and then go through the large non-polar portion of the membrane. Example is photosynthesis and cell respiration. And we have facilitated diffusion, which uses large polar or charged molecules like glucose, and they need to use this as they are too big to get between phosphate heads and would not be able to pass through the large non-polar region of the cellular membrane. Each type of or class of molecule will have its own protein channel to fit its shape. Example, glucose in the bloodstream after eating. Then there's primary active transport, which is the large polar or charged molecules using the specially shaped channels. But in this case, the chemicals are moving against a concentration gradient. So its example is glucose being reabsorbed in the kidneys. Then we have endo or exocytosis, which involve very large molecules or particles that cannot diffuse into or fit into a channel. So the example is pinocytosis, so very large molecules like DNA that are in one solution. Then we have the details of osmosis. So first, there's a hypertonic solution, which is the fact that outside of the cell is more concentrated than inside the cell. So this is the movement of water molecules outside of a cell. It results in the shrinking of a cell. So more solute in the exterior environment than H2O. Solute cannot cross membrane with ease. They use facilitated diffusion or something more complicated than diffusion. Water molecules are more likely to move to even out the concentration, so H2O molecules move out of the cell, shrinking it. Then we have hypotonic solution, which is outside of the cell is very low in solute, more H2O molecules than solute on the outside. And then inside the cell, there are more solute than H2O molecules, so water molecules are going to move into the cell from outside of the cell. This will cause the cell to expand and maybe even burst if it's an animal cell. In plants, they won't burst. Then we have the isotonic solution, which is when the concentration of the solute to water is the same both inside and out, so it's the ideal situation for a cell. H2O can move out and in at the same rate, and the size of the cell never changes at any point in time. Then we have enzymes. So what are they? There's protein catalysts, which speed up chemical reactions without being consumed, and their names often end in "ase." Why are they necessary? They are necessary because reactant molecules must collide with enough force or energy and with the correct geometric orientation for bond breaking to occur. This happens in a random and uncontrolled manner in non-catalyzed environments. Where does the energy required for chemical reactions come from? Most chemical reactions get their energy from heat. However, living cells can't rely on heat as a source of activation energy due to denaturation. The purpose of an enzyme in a cell is to allow the cell to carry out a chemical reaction as quick as possible without using too much energy. Enzymes do this by lowering the activation energy. Enzyme structure. Enzymes overcome the activation energy barrier by holding chemical reactions in the correct orientation. A substrate is the reactant that an enzyme acts on. It binds to a particular site on the enzyme called the active site. Enzymes can change their shape slightly to accommodate the substrate, but substrates fit closely into active sites. The adjustment in shape is referred to as induced fit and forms the enzyme-substrate complex. And enzymes work best in enzyme catalyst in saturated, and it has an optimal pH and temp that it works at due to particle theory and denaturation. Some enzymes also require non-protein cofactors such as inorganic substances or organic coenzymes before they can work properly. So some things that inhibit enzymes, the inhibitors are chemicals that bind to specific enzymes, which result in the change of the shape of the enzyme, which shuts it down. There are two main types, competitive and non-competitive. Non-competitive inhibition is when an inhibitor molecule binds to the enzyme at a site known as the allosteric site. As a result, the 3D structure of the enzyme is changed, which prevents the substrate from binding to the active site. Molecules that promote the action of enzymes can also bind to the allosteric site. They are called activators. Competitive inhibition is what involves chemical compounds that bind to the active site of the enzyme and inhibit enzymatic reactions. The compounds compete with the true substrate for access to the active site. An example is penicillin. Industrial uses of enzymes are the starch processing industry, dairy industry to create cheese, and cleaning industry, so allow stains to be removed at lower temperature and less mechanical agitation.